Welcome to Chapter 3, Energy, Catalysts, and Biosynthesis. This is the last of our review chapters. Once again, this lecture will be relatively brief. Most of this information should be a review to you from previous chemistry classes. In today's lecture, we're going to discuss how cells use energy, enzymes and how they work, properties of energy, and biosynthesis. And we're going to go over what I want you to learn from this chapter. So go ahead and read through this list. You can pause the video to do that. I won't read it to you. Let's start our discussion of Chapter 3 with how cells use energy. And under this topic, we're going to talk about catabolic and anabolic pathways, biology and the laws of thermodynamics, where does all this energy come from, and how energy is stored. Cells are always completing both anabolic and catabolic pathways. Catabolic pathways are those that break down molecules such as food into usable building blocks such as amino acids. Conversely, anabolic pathways work to create new macromolecules and structures from these building blocks. An easy way to remember these two pathways is A, B, C, D. Anabolic builds, catabolic destroys. Energy release is also an important aspect of these pathways. Besides creating building blocks for the cell, catabolic pathways also release useful energy such as ATP and energy in the form of heat. Once again, conversely, anabolic pathways need this energy to create these molecules. Next, let's discuss biology and the laws of thermodynamics. We can never escape physics no matter how hard we try. Just like all other natural phenomena, biology answers to the two laws of thermodynamics. In summary, the first law states that energy cannot be created or destroyed, only transferred. This is the principle that the carbon cycle is built on. Plants harness solar energy and use carbon dioxide to store it as usable energy in carbohydrates. We then consume these foods and release the energy from these molecules for our own use and release the carbon dioxide back into the environment for further utilization. However, not every system is perfect and heat is lost during these transfers. That's where the second law comes into play. The second law, in simplified terms, states that all systems will move toward a state of disorder. We measure this disorder as entropy. At first, this law seems to contradict everything we know about biological organisms. We are highly organized organisms. So based on what we know about the cell and biological organisms, how can biology exist in the same space as these two laws? This is where heat balances the two laws. As we become more ordered, we put off heat into the area around us, creating a greater amount of entropy or disorder in the universe. The cell must create as much or more entropy than the order it is creating. The heat produced from cellular processes helps create this balance. I've already mentioned this, but it can't be mentioned enough. All energy on Earth is made into usable energy through photosynthesis from solar energy. We'll discuss this more in Chapter 13, but here is an overview. Photosynthesis occurs in two stages. The first stage is the light reactions, where the solar energy is utilized to create energy carriers that then transfer this energy to the Calvin cycle, where sugars are created from CO2 and other molecules. These sugars are then stored as carbohydrates in the plant or are used to make proteins, nucleic acids, and lipids. Anything the plant needs. Plants are known as autotrophs because they can generate their own food. Since we cannot generate our own energy, we get our energy from plants or from animals who have eaten plants. Next, let's go over how energy is stored. Within sugars or other macromolecules, This usable energy is stored in the bonds of the food item. To release this energy, living organisms go through a process known as respiration, where the molecules are oxidized to release their energy to create activated energy carriers. We will discuss these activated energy carriers later in this lecture. I've loaded some refreshers on redox reactions on Blackboard to refresh your memories. But here is my favorite way to remember it. Oil rig. Oxidation is losing an electron. Reduction is gaining an electron. We've briefly referred to enzymes in this course already, and you should be relatively familiar with them from previous biology courses. But let's spend some time reminding ourselves of what they are and how they work. The topics we will be covering are what are enzymes and how enzymes lower activation energy. Enzymes, for the most part, are proteins that have folded in a specific way to create a unique or specific active site. This active site binds with a specific substrate and helps catalyze a reaction to occur, generating a new product. This picture simplifies the process. Cells have many types of enzymes, all created for specific purpose in the cell. Notice how I keep using the term specific? That is because each enzyme, an enzyme family, can only perform a specific function. These functions have evolved along with us throughout history. In fact, many evolution-based experiments have focused on the similarities of enzymes between organisms. One current question circling the scientific atmosphere is the ability for adult humans to catabolize lactose and why this is a fairly unique trait among mammals. All reactions have an activation barrier, or the energy it takes to break a covalent bond. Some of these reactions have a very low barrier and will proceed with very little energy. These reactions are known as spontaneous, not because they happen quickly, but because they happen without much energy input into the system. An example of this is an avalanche. It takes very little to start an avalanche because the delta G is negative. However, other reactions have a high activation barrier and thus a positive delta G. Enzymes will modify the conformation of the substrate to create a more favorable conformation for the reaction to occur. Thus, enzymes either speed up or allow reactions to proceed without being consumed by the reaction. We will discuss enzymes more in Chapter 4, but before we move on, we need to address two important mathematical barriers, Km and Vmax. Please note that these are not the only variables associated with the properties of enzymes, but two of the most basic we need to address. Enzymes must bind with substrates to create reactions. Typically, the rate of turnover is limited by the amount of substrate present. However, when Vmax is reached, all the active sites in the enzymes are bound to substrate, thus making the limiting factor in the reaction of the product the turnover rate and not the availability of substrate. This is Vmax. This Vmax, in other words, tells us exactly how fast the enzyme is capable of functioning. Think about this in terms of the fermentation experiment we performed in the Biology 1 lab. By adding the amount of lactase we did to the yeast suspension, we were able to hit Vmax immediately by providing more lactose than lactase present in the cells. The rate of fermentation we then observed was limited by the turnover rate of the lactase enzyme. Km, on the other hand, is the other part of an important equation known as the Michaelis-Menten equation. Km is an indication of how much substrate is needed to reach half Vmax. Using our yeast example, the amount of lactose it would take to reach half the maximum rate of CO2 production would be the Km. This value is especially helpful when trying to determine the parameters of an experiment. Well, that's about it for now. I've decided to split this topic into two So give yourself a break, come back, and start part 2.